Hi everyone, my name is Charlie. I'm an ophthalmology resident in the DeAngelis lab at the University of Buffalo, and it's my privilege today to join you all to discuss our research.
Can you please tell us about the research presented here at ARVO?
So our lab, the DeAngelis lab, focuses on AMD research, and as many of you know, AMD is a very common disease that is a major leading cause of blindness. And although there are drugs that we have to treat the disease, and we know certain risk factors such as age and family history, there's still a lot about the underlying pathogenesis in terms of the disease progression. So one of the major hurdles that we face collectively as AMD scientists is that we don't have any animal models that can fully recapitulate the full spectrum of this disease, nor do we have cellular models that can capture the complexity of the architecture and cellular function of the human macula. The way that the DeAngelis lab approaches this problem is, rather than looking at these other models, we look directly at the human tissue that's affected by disease. That is, the macula and the retina and RPE of deceased donor eyes. What we do is we have this, Dr. DeAngelis established this thing called the Utah Protocol, where we rapidly autopsy these eyes within 6 hours with a death to preservation time that helps us ensure RNA integrity as well as minimize postmortem changes. And all these things occur within 6 hours. During this time, we also want to make sure we accurately phenotype these eyes. So what we do is we do high-resolution Heidelberg SD-OCT as well as fundus imaging, and we send all this information to a team of clinical retina specialists, and they have to reach a consensus on the correct phenotype, whether this is a normal eye, an intermediate AMD eye, or an advanced AMD eye. And then finally, we will meticulously dissect these eyes to separate out just the macula of the affected tissue, either at the level of the RPE or the retina. The work that we presented is focusing primarily on bulk RNA polytail enhanced sequencing. So what we did is we took some control eyes, intermediate and neovascular eyes, did the RNA sequencing, and then the resultant data was then subjected to statistical and bioinformatic analysis using PCA, DEC2, Bonferroni correction, as well as Benjamini-Hochberg analysis. And what this allows us to do is to look at the differentially expressed genes, or DEGs, between disease states, as well as identify pathways that show increased or decreased expression that could be associated or protective against different disease states. One of the major takeaways that we get from this data is the importance of specifically the RPE in AMD pathogenesis, especially when we look at things at the bulk RNA level. Firstly, when we look at these differentially expressed genes just in normal tissue, what we did is we went back to the seminal paper by Frichte et al., and we looked at all the identified GWAS loci and determined whether or not there was differential expression between the retina and the RPE. What we found is that of the GWAS that were identified, 75% of them actually had showed differential expression between the RPE and the retina. But furthermore, of those 75%, 75% showed greater expression within the RPE itself, again, pointing to the importance of this tissue in AMD pathogenesis. When we look at the disease states, we see an even greater difference between the asymmetry in the differentially expressed genes. For example, I believe in neovascular disease versus normal, or neovascular versus intermediate AMD, we found around maybe 40 to 50 differentially expressed genes within the retina, whereas when we look at the RPE, we see around 1,200 differentially expressed genes. The last step that we actually showed in the presentation is when we perform ingenuity pathway analysis, or IPA, which allows us to evaluate the common canonical pathways that govern cellular function. We see that, similar to the DEGs, we see a great asymmetry between the retina and the RPE choroid. We found that although there were something like 21 canonical pathways that showed differences between intermediate and control, and I think it was 60 or 62 different pathways between neovascular and control, and neovascular and AREDS2, a majority of these pathways occur within the RPE choroid. Additionally, when we look at these pathways, what we found is that the pathways that showed differential expression in the retina really only had 1 or 2 genes that showed differential expression, whereas in the RPE choroid, there was an average of 14.5 genes within each of the 55 identified pathways, and this comprised as much as 54% of the entire pathway. Finally, what we wanted to do, and one of our major questions, is how does this kind of fit into everything? Like, how does this relate to disease pathogenesis? And although there's a lot of literature about different risk factors for AMD, what genes contribute to AMD risk, and our lab is also extensively published on these things, there isn't that much literature out there about what genes are, what pathways are protective against neovascular AMD. And yesterday, what I presented was focusing in on the genes and the pathways that were found to be protective against neovascular disease. That is, when we looked at expression between AREDS3 and neovascular disease, we found that there was greater expression in AREDS3. So what we found is that this one pathway, the axonal guidance signaling pathway, we found a high number, 7 genes that were found to be protective against neovascular disease. And using IP analysis, we can actually kind of look to see in the grand scheme, where do these pathways, where do these genes fit together, and under which pathways do they work? And we found that BMP signaling and Wnt signaling were highly protective against neovascular disease, as well as that some of the downstream effects of the Wnt signaling pathway, when perturbed, increase the risk of neovascular disease. The reason why this is very exciting, though, is that this really corroborates a recent publication that we had just published last week in Cell Genomics, where we looked at, instead of the bulk level sequencing, we looked at single cell sequencing of the RPE. And in that type of analysis, what we found, similar to us, is that the frizzled Wnt pathway is perturbed in advanced stages of AMD, suggesting that it does possibly play a role in AMD pathogenesis. And these findings were also shown in that particular study to be associated with chromatin changes, which suggests that there are epigenetic contributions to that lead to the importance of this pathway, which, again, as AMD scientists, we've always kind of thought that, you know, it's the genetics as well as the environment through epigenetic means that underscore the entire pathogenesis of the disease.
Can you tell us the relevance of processing the eye tissue within 6 hours of death?
That's an excellent question. So one of the big problems is that when you are looking at cadaveric tissue, there's always the chance that there's going to be degradation. You know, inside the human cell, we have DNA that stores the genetic information, but then also RNA, which forms a copy of this information for the production of proteins, and these proteins are really what govern cellular function. So in the grand scheme of things, though, DNA is considered long-term storage, and RNA is something that's temporary, that's used by the cell only within a short milieu before it's turned into a protein. So within our cell, we actually have a lot of endonucleases that are known to digest RNA. And one of the nice things is that in this particular study, we're looking at mRNA, which has a poly-A tail, which is thought to protect the degradation against it. However, the longer you wait, the more time that you can have degradation of the RNA molecules. So one of the reasons why we want to make sure that we do things as fast as possible, that is within a 6-hour window between not just the time that we nucleate, but rather the time that there's the pronouncement, is because we know that there is going to be RNA degradation. And one of the very important things is that we can't assume that RNA degradation occurs at the same rate between normal tissue and diseased tissue. And you know, if you let this go on for too long, there's a chance that you can create artifactual changes that don't represent what's truly going on within the eye. We actually showed in our Utah protocol, using several different RNA integrity kits, that our RNA quality is actually very high. And additionally, what we were able to demonstrate in that similar study is that we had very good tissue separation. What we do in our dissection process and our validation process is we look at genes that are specifically expressed either just in the RPE or just in the retina, and we look at the expression levels of these genes in our 2 different dissected tissues. What we would expect is that in the RPE tissue, you should see those genes that are only supposed to be expressed in the RPE, but not within the retina. And that's almost exactly what we found. We found that these genes, we selected a couple of them, had something like represented 4 or 5% of the total number of copies that we found in the RPE, and something like 0.003% within the retina, which again just demonstrates the quality of our RNA as well as the fidelity of our tissue separation.
Is the Utah protocol used in all genetic testing, or is this protocol primarily done in the DeAngelis lab?
I believe that this is something that Dr. Meg DeAngelis developed herself with the help of Dr. Leah Owen, and it is something that we have published on, and it's something that we feel is very important in terms of that demonstrates the quality of our tissue repository. To the best of my knowledge, Dr. DeAngelis is the one who kind of pioneered this and is the primary person who's performing this type of, this quality of tissue dissection and preservation. But there could be others that are working across the U.S. that may be adopting these changes in their donor eye repositories as well, and we'd be happy if they had any other questions on any technical issues on how to do this.
By identifying the protective gene pathways in AMD, can this translate into possible treatment options?
Well, that is also an excellent question. As many of you know, right now for AMD, we don't have that many treatments. For the last 20 years, when it comes to AMD, there's intermediate disease and neovascular disease. And the only thing that we really have to kind of reduce risk of progression from intermediate to advanced disease are the AREDS2 vitamins that was demonstrated in the AREDS trials. However, this really only reduces the risk of progression by around 25%. When they do convert to advanced disease, if it's neovascular disease, we do have anti-VEGF injections, and this has been around for around 20 years now. Recently, there was an FDA approval for certain complement inhibitor for the treatment of geographic atrophy. However, again, one of the important things is that other than the AREDS vitamins, both the intravitreal injection of the anti-VEGF medication or the complement inhibitors are there only once you reach the advanced disease. And really, it's in the advanced disease, you've already lost a lot of vision. One of the things that our lab really wants to evaluate is that transition between intermediate disease and neovascular disease. And, as I mentioned earlier, we did find that upregulation of the BMP pathway and the Wnt pathway are associated with protection against the progression of this advanced disease. And what we're hoping to do is to identify targets so that in future studies we can look to see, is there anything that we can target in these pathways to, rather than just treat the disease once it becomes advanced, but rather protect against even reaching those advanced stages of AMD. Of course, the work that we do at the genetic level and the genomic level really is there to help generate hypotheses, and further studies at the functional level kind of looking at specifically how these pathways interact with each other within the cellular milieu of the macula are needed so that we can develop future therapies.
So what are your next steps in this line of research?
So one of the next steps for us is we actually recently got a PhD student who joined our lab who has a lot of experience with functional work, looking at proteins and looking at, beyond what happens just at the RNA level. So what we hope to do really is to, again, leveraging the tissue that we have, although I did mention that, we get the genetic information, we also have a lot of other information from these patients. We have samples of the vitreous, we have samples from the serum, we have samples of the actual eye itself. We can kind of look at the next steps. That is, normally it goes from DNA, RNA to protein. So we want to start looking at the protein level. What are these changes? What do the changes in the BMP and the Wnt pathway cause in terms of protein upregulation, protein downregulation, and how do these specifically interact with each other to cause the neovascular phenotype that we see in these eyes?
I see that a portion of this research is being funded by Genentech. Are they working on a possible treatment using this line of research?
So I can't speak directly as to, what the Genentech people are doing outside of the research with us, but, we are very fortunate to be able to work with Genentech on this type of research because, with their resources, we're able to look at single cell data, which allows us not only to corroborate the findings that we see at the bulk RNA level, but also to look at the different cell types and how they change in disease states. Like I mentioned, in our RPE, we are able to find changes, but we didn't find significant changes in the retina. One of the hypotheses behind why this occurs is that in the normal human macula, we, in collaboration with Dr. Ray Chen at Baylor, have found that there are over 70 different cell types in terms of the phenotypes that we see at the single cell level within the human macula, and that it's most likely needed, single cell techniques are most likely needed to determine within the macular retina itself what are the different changes that occur between disease states. So working with Genentech, what they've done is kind of helped establish what changes occur within the retina at the single cell level and as well as the RPE. And then using this, we can determine, what specific targets and what specific cells we can potentially use as druggable targets in development of treatments for AMD. Of course, all of our work is very early right now, but that doesn't mean that we're not excited because we have many new, groundbreaking findings that will be presented by Genentech later today.
Thank you so much for sitting down with us today and sharing this exciting breakthrough in research.
I really want to thank the Macular Degeneration Foundation, not only for giving me this opportunity, but for all their support in our research.